In this 3-mark subjective question, they are directing us to illustrate the usage of radioactive sulfur and phosphorus to represent the DNA is a genetic material by Arsha and Chase. For this narration, we are illustrating the diagram given as per the CBSE curriculum for mentioning each one of the process. In the first segment, we are growing the bacteriophage in a 13 sulfur nutrition medium which is marked as red, whereas in the second case we are growing the bacteriophage rich in 32P, so that the bacteria makes the DNA by using 32P. The other bacteriophage is using 35S for making its protein. So both the bacteriophage are allowed to infect the bacteria. In the infection stage, once the bacteriophage is infecting the bacteria, the DNA is getting entry into the bacteria. In the blending stage, we are removing the bacteria and phage away from each other. When you are doing the centrifugation, we come to know the bacteria having the radio-labeled DNA or unlabeled DNA after examining under autoradiography. We are finally finding out the radioactive protein is present in the bacteriophage, whereas the bacteria is having radioactive DNA. This is exactly illustrating only the DNA is responsible for transferring the genetic material. In this way, if you are mentioning, we can get 3 mark. For infection mention, we will be getting 1 mark. For blending mention, we will be getting 1 mark. For centrifugation mention, we will be getting 1 mark with the appropriate narration, so that we can satisfy the total 3 mark demand of this subjective question. In this 3 mark subjective question, they are directing us to specify the way how Meslin and Stahl experiment was designed to prove semi-conservative mode of DNA replication using nitrogen isotope. For this, we given the NCERT specific semi-conservative mode of DNA replication proof. In this particular picture, the first step is illustrating when we are growing the E. coli in 15N nutrition medium, all the bacteria is making the 15N DNA that we are isolating and sedimenting. While sedimenting, we are observing the band is getting segregated at the lower base, indicating the weight is maximum. The same bacteria we are growing in the 14N nutrition medium, we are observing the DNA which is formed in the middle, not either in the extreme top corner or in the bottom corner. We are observing the middle, so it is illustrating the DNA is having both the 15N and 14N DNA. And we are making the E. coli to grow in the second generation in the 14N nitrogen isotope alone. When we are growing, the E. coli is making the 2 type of DNA. One DNA is having only 14N DNA, other DNA is having both 14N and 15N DNA. With this information, we are specifying the semi-conservative mode of DNA replication after observing the DNA band segregation in the top corner and in the middle corner, which is representing the semi-conservative mode of DNA replication happens by using the parental strand to synthesize a daughter strand. In this way, if you are illustrating, we are getting the 3 mark for this subjective question.